Hi, welcome to the Bioinformatics Chat. Today I'm speaking to William Towns. William is a postdoc at Princeton University, and we will be talking about his method of dimensionality reduction for single-cell RNA-seq data. Will, welcome to the podcast.
Thank you, Roman. It's a pleasure to talk to you.
Tell us how you became interested in single-cell RNA sequencing, and how did you notice that something is missing in this very crowded space of single-cell RNA-seq analysis?
Sure. Well, first of all, I had a very windy path to bioinformatics and biostatistics. My PhD is in biostatistics. I started out studying tropical ecology and botany and shifted into software testing and worked for about 6 years as a contractor for the Environmental Protection Agency. Then I ended up, deciding to make a change and pursue a PhD and focus on biomedical data analysis and biostatistics. But I never thought that I would be able to actually do research or, complete my program. I just saw it as a way to get paid to learn some cool new ideas and to meet some interesting, smart people. So I was shocked that I actually passed the qualifying exam and had to choose a thesis topic. And I did not know what RNA-seq was when I got into grad school. In fact, I took a class in undergrad called Modern Genetic Analysis. And the hottest topic was the human sequencing of the human genome. And we barely touched on microarrays. So that was about 2006 or 2007 when I graduated from college. So you can imagine, having been away from biology for several years, coming in and hearing about ChIP-seq and RNA-seq and single cell, and CRISPR was coming out, about halfway through my PhD. So it was just kind of overwhelming. So I was very lucky to do a rotation with a professor named Martin Ari, who is primarily in the Massachusetts General Hospital. He was really a great person to work with, kind of introduced me to single-cell RNA-seq and got me playing with some data, doing some exploratory and looking at just really quality control issues and very basic stuff. And then he introduced me to Rafa Irizarry, who eventually became my thesis advisor. And at the time, Rafa had a postdoc named Stephanie Hicks, who I started working with very closely. And Stephanie was really a great mentor for me and helped me believe that I actually could make a contribution to what's a pretty fast-moving field. And she helped me get up to speed and helped me think of some interesting topics. And she had been studying this phenomenon of the correlation between the first principal component and the fraction of zeros.
Let's explain it in a bit more detail. So she was performing PCA, right, on single-cell RNA-seq data. So how does that work and what's it used for? And maybe explain a bit more about that correlation.
Sure. Well, the standard procedure around that time, I would say that was probably 2015, 2016. Most of the single-cell RNA-seq data were from protocols like SMART-seq, where you don't have unique molecular identifiers. You don't have UMIs. So you're looking at read counts from a relatively small number of cells, and the sequencing is fairly deep. So you would detect a lot more genes in these data compared to like a 10x droplet data set. So what she was doing was just basically searching for sources of bias and systematic biases, because I really don't know why she decided to search for these biases. I think basically Rafa just really didn't trust a lot of the results that were coming out from these data because they are so noisy. And he, I think, just had a hunch maybe that we should start digging around and seeing if there's something amiss. And Stephanie identified, she looked at a large number of data sets, public data, of course. And if you follow the standard procedure, which is basically you have these counts, you're going to normalize them somehow. Maybe you convert them to transcripts per million, which is basically you divide the count for each gene by within a cell by the total counts of the cell. So you kind of convert it to a proportion and then multiply by a million. That's counts per million, but you could also sort of adjust for the gene length, especially with a full-length protocol like SmartSeq. You want to take care of any bias, like longer transcripts being more likely to be overrepresented and things, GC bias and stuff like that. So if you have those normalized units like CPM or TPM, by the way, those normalizations came from bulk RNA-seq, so they were around for a long time. Then people want to apply PCA, but if you just straightforwardly apply PCA, PCA stands for Principal Components Analysis, and that is a very fast and popular method for reducing the dimensionality from 20,000 genes or whatever to maybe you just reduce it to 2 dimensions so you can make a nice plot, like a scatter plot of all the cells, or you could reduce it to 10 or 50 dimensions if you want to do some clustering on the low-dimensional space. Even, by the way, t-SNE, t-distributed stochastic neighbor embedding, actually the standard implementation does a pre-dimension reduction down to about 50 PCs before it even starts to run the actual t-SNE algorithm. So PCA is just everywhere.
And the simple intuition behind PCA is that if you imagine your cells, right, lying in this high dimensional, like 20,000 dimensional space, you find a low dimensional space and sort of just geometrically project these points onto a, let's say, plane or hyperplane. And PCA is basically finding such a plane that this projection is the most informative, right, you would say?
Yeah, absolutely. And the key question is, what do you mean by informative? And the way PCA mathematically defines that is just simply using a Euclidean distance. So you look at sort of the sum of the squared Euclidean distances between the actual points in the high dimensional space and the projection of the point onto the low dimensional subspace. And, then it's simply an optimization problem where you want to minimize this mean squared error or sum of squared errors. And it's really a sort of Euclidean distance metric that is used to define most informative.
And so you were saying that Stephanie ran PCA, right? And she noticed some correlations that she didn't perhaps expect to notice?
Yeah, exactly. So every cell has the number of genes in the cell that are zero versus non-zero is generally not thought to be biologically interesting. There may be some special cases like perhaps with developmental processes where you do expect stem cells to have just a larger number of genes active than a very specialized cell type that's further down the trajectory. But if you're just looking at just a generic tissue and you want to find subtypes of cells, you're probably more interested in finding marker genes that kind of tag a certain cell type rather than just saying, oh, this is a cell type A where all the genes are turned on and cell type B has a fewer number of genes turned on. We don't care about the identity of the genes that are on or off. So that is kind of we would consider it sort of a nuisance. It's a statistic that represents a kind of nuisance variation. So when you apply normalization, your goal is to sort of remove a lot of those nuisance variations, and so that they won't contaminate the low dimensional projection and things like differential expression and so on. But unfortunately, even with normalized data, and by the way, she's also doing the standard thing of taking the log of 1 plus the counts, 1 plus the normalized counts. That was very widespread and still widespread transformation because of the skewness of the. And you have to add the pseudo count of 1 because you have so many exact zeros. You can't just take the log of 0 because that's minus infinity.
Right. So the intuition behind that is that PCA is sort of designed for normally distributed data. And so if your data doesn't look normally distributed, then you have to massage it in some ways to make it look like normally distributed. And so the log transformation is like commonly used for that for no good reason, right? Just because it happens to seem to work.
I think it works with bulk RNA-seq because you don't have very many exact zeros. But there are many other transformations that have been considered, variance stabilizing transformations and Anscombe. Well, I think that's the same thing, the Anscombe transformation. But the bottom line is all of these transformations are based on an asymptotic method called the delta method. And you assume a certain sampling distribution, and then you sort of you can get the mean and the variance of different transformations of that data. And you want to find a transformation that makes it so that the variance is independent of the mean. So the normal distribution is one of the only distributions that the variance is not a function of the mean. And that removing that relationship, like you say, it makes the data more friendly for methods like PCA that are implicitly based on normal errors. But what Stephanie found when she actually applied this, what I call the standard pipeline, was you would see the first principal component, which is the dominant axis of variation in the data, was highly correlated with just the fraction of genes that were 0. And it's a very troubling result because it suggests that if you just run clustering on your low-dimensional space, you might find clusters that really have nothing to do with biology, or it might inhibit, even if the biology is really strong, it might sort of muddy the waters and make it harder to be clear about what are different cell types and so on. So that was really the motivation for my work, was to try to find an alternative to the so-called standard pipeline that would avoid these spurious, sources of variation showing up in the low-dimensional space.
And I think one take on this phenomenon that I've heard is that people are basically aware of this effect, and so what they would say is, just drop that first principal component, because you have a lot of principal components. There's no shortage of them, and if you know that this principal component is responsible for something that you don't care about, you just drop it and use, for example, the second, the third, and the fourth principal components. So one might think that that's actually, PCA working as it should. So it gives you a number of uncorrelated variables, new variables that describe your data set. And you just try to reverse engineer what those variables mean in terms of the biology of your experiment. And then you pick the ones that are interesting. But that wasn't your take, right? You saw this as a problem with the method itself.
Sure. Well, what you just suggested is actually the first thing that I tried. And it does work in the sense that if the technical we were calling that at the time the detection rate. So the detection rate just meaning for each cell, what is the fraction of genes that are 0? the detection rate is 1 minus the zero fraction. So how many genes were non-zero? The first thing I did was that, okay, if this is correlated with principal component number 1, we know that PC2 is orthogonal to PC1 and so on down the line. So let's just, like you say, let's just sort of project off the bad part of the subspace and, only focus on PCs 2 through, 7 or whatever. But the problem with that is sometimes the detection rate is actually correlated with all of the top 5 principal components. Every data set is different. So in some data sets, in fact, the biology is very strong, and biology is PC1, and then this noisy stuff is coming in through PCs 2 and 3, and it's not a universal rule that it's always the first PC. The point is more that it's in there, kind of messing things up, and it doesn't always perfectly match with one of the PCs that's removable. So the next, approach that we took to get around that was, you can go into sort of the theory of linear algebra. You can say you've got this matrix of all these PCs, and you can actually do a projection onto the orthogonal complement, I guess is the technical name. You basically say that there's a subspace defined by this vector of detection rate, and there's the subspace that we actually have, and we want to sort of partition it into 2 orthogonal parts, and then only look at the part that will force the correlation between the resulting adjusted PCs to have zero correlation with the thing that you don't want. And what it really boils down to is you're just regressing off. It's equivalent to doing a linear regression. In fact, you can regress it off from the original data set and it'll have the same result.
Right. So you're sort of recognizing that the PCA is not perfect. It has these flaws, and none of the principal components exactly captures this nuisance parameter. So you guide it, right? You force it to ignore the nuisance parameter.
Yes. So we tried that, and it sort of solved the problem of the detection rate, mathematically, right? But it was still bothering me a lot that nobody's really given a good justification of, this log transformation, why we're putting in a pseudo count of 1, all of that kind of stuff. Why do we go through all these gymnastics to make the data, fit with PCA when, let's face it, the data is just not normally distributed. If you look at any histogram of single cell, it just really doesn't look at all remotely like a normal distribution, even after you do all the, transformations and so on.
Yeah, and one obvious tell that it's not normally distributed is that it's discrete. So
yes,
strictly speaking, it cannot be normally distributed. It cannot be generated by a sampling from normal distribution.
Right. But I would say that bulk RNA-seq is also discrete, but linear Gaussian methods for things like differential expression are somewhat more successful with bulk RNA-seq than single cell. And I think it's because when you have a discrete distribution like a Poisson or a negative binomial, if the mean of that distribution gets really large, if the counts overall are pretty high and you don't have that many zeros and ones, the discreteness starts to kind of blur away. And there is, it's really just the central limit theorem that shows that a Poisson with a really large mean, it really could be well approximated by a Gaussian. So, there is some justification if the counts are really high, then probably PCA would do just fine. And you could do your log transformation and adding a pseudo count of 1 won't matter because all the counts are really large. But if you have data, like the single cell data where the vast majority, like 80, 90 or more % of the genes are zero, and then you have a bunch of 1s and a bunch of 2s, and maybe you might see a 15 or a 20. Actually, I'm talking about UMI counts now, but the same basic idea goes with read counts. You have tons of zeros and low counts. Then that pseudo count really makes a huge distortion between all the zeros and all of the non-zeros. So it kind of introduces this artificial bimodality, which is really a result of the transformation and normalization as opposed to the data itself. So that sort of spurred us to think, can we just get rid of the whole apparatus and directly do dimension reduction on the raw counts instead of trying to finesse things to make them fit the PCA desired input.
And while looking at the data, right, you noticed that there are different types of data sets, and they have different statistical properties. And we're talking about the presence or absence of unique molecular identifiers. So can you explain what UMIs are and how exactly they affect the statistical properties of the data set?
Sure. So early single-cell RNA-seq data mostly did not have unique molecular identifiers, or UMIs. There were some UMI data early on, but I think they didn't really take off until the droplet protocols like DropSeq, InDrops, and especially 10x genomics, which came a little later, allowing people to analyze thousands of cells instead of hundreds. But what a UMI is basically just a little barcode that that identifies what molecule, what unique molecule different reads originated from. So read counts are the end result of polymerase chain reaction, or PCR, which takes the small amount of RNA, or really cDNA, complementary DNA, which comes from RNA, and amplifies it to a large enough quantity that can be sequenced, because these single cells have such a small amount of starting material. So what that does is it takes the original molecules and it just copies them over and over and over again. So you could end up with a situation where a particular gene or transcript is represented by hundreds of read counts that all originated from, one or two or three actual molecules. So the PCR really distorts the data quite a bit. And being able to remove those PCR duplicates is I consider a major innovation in single-cell RNA-seq. And I just really want to give a shout-out to the hardworking folks in the wet labs all over the place who are really doing great innovation. And I think their innovation matters a lot more sometimes than these computational tricks that people like me are trying to come up with. So I have a lot of respect for the work they're doing. And I'm very grateful that they were able to make the data so much cleaner with the invention of these UMIs. So the characteristics of read count data are you still have a lot of zeros, because if you have zero molecules captured or at the end of the reverse transcriptase step, or just that there were zero molecules in the original cell, no matter how many times you multiply zero, you still have zero. So the zeros are not affected by PCR, but all the non-zero stuff, the ones and the twos, get amplified to be really large. So if you look at read count data, such as that from SmartSeq 2 or any number of other read count protocols, they really look zero-inflated. You see this sort of distribution that's sort of a log normal or maybe a negative binomial or something, and then that's the non-zero component, and then you have a big spike at zero. So historically, a lot of the methods developers focused on these sort of zero-inflated models, and I myself also developed a zero-inflated, very computationally complex Bayesian hierarchical model that we called varying censoring aware matrix factorization, which was just really clunky and it was too much. But we felt like we needed to have all of these zero inflation mechanisms to address the big spike at zero in the read counts. But after that, Riso and several others came out with ZINB Wave, which was a much better implemented dimension reduction that included zero inflation. And after we saw the ZINB wave paper, we felt like, well, we really should just abandon this VAMP method because they've done a much better job. And we really learned a lot from that paper. They also were very creative to find a way to include covariates as well as dimension reduction in a simultaneous model, which was really impressive. But we started looking at the newer data sets, the unique molecular identifier counts, and we noticed that first, they still had the problem of the correlation between detection rate and the first PC if you analyzed it with principal component analysis. So Stephanie's insight was still valid for the UMI counts. But what I noticed was if you look at the distributions, they don't really have the zero inflation. They just sort of look like a over-dispersed Poisson. And the easiest way to see it was to look at negative control data. And just as we were starting to look into these negative control data sets where you just have spike-ins or you just have one purified cell type and you don't expect any heterogeneity due to different cell types, Valentine Svensson came out with a really influential blog post where he claimed that droplet single-cell RNA-seq, which implicitly has unique molecular identifiers, was not zero-inflated. And it was a very, I want to say, controversial blog post, and he convinced me 100%. I found it very persuasive, and I think he did a great job.
I think that blog post recently got published in a journal. If people thought that blog posts don't count, well then you can actually cite that paper in your papers.
Yes, and I'm proud to say I may have had a very small role in that. I was not a reviewer or anything, but when we started to write up our paper for publication, I wanted to cite Valentine's blog post, but we didn't really know if the journal would accept a blog post citation. So I contacted Valentine and asked him to please convert it into a bioRxiv preprint so that I could at least have something to cite. And I don't know if that's what led down the path to it getting published, but I'm
sure it did.
I'm happy that he got the recognition for it that he deserves. So, yeah. So I think that pretty much summarizes the problem that we were trying to solve.
Okay, and so if we don't have zero inflation for UMI data, then we could apply something like ZINB wave that you mentioned, right? And just maybe disable that zero inflation component there, right?
Yes, that's right. So ZNB wave is a very general model. It's very sophisticated and has a lot of parameters. So for a while, I thought, I don't really need to work on this problem anymore because ZINB wave has solved everything. But there's just a couple of issues with ZINB wave. First of all, it's rather slow. And I think part of the slowness was due to the fact that it's trying to solve the problem of dimension reduction for both the read counts and the UMI counts. So it's sort of this omnibus approach. And we really felt that we could make some at least incremental improvements by focusing just on UMI counts, because those were becoming very popular. And we thought that we could come up with a simpler algorithm that would be a lot more scalable. And another concern that we had was the factors that ZINB wave was producing are not really as interpretable as the factors from principal component analysis. And the reason is because they have two different loadings matrices. And the way to order these latent dimensions in a linear dimension reduction like ZINB wave is you want to sort of rotate the loadings and make them orthonormal. And then that makes it so that the the factors are sort of in decreasing order of importance. But ZNB wave was not able to do that because of their sort of double likelihood. So that was another area we felt that we could make a contribution and give a result that took the crucial aspects of ZNB wave, but gave people a result that was a lot faster and more interpretable in a way very similar to PCA. So I really want to give credit to Riso et al. Because ZNB wave was a major, major innovation. But we did decide to try our hand at writing our own dimension reduction. And I think the accuracy of ZNB waves never really in question, but it's just a little bit slow and not quite as interpretable. But it is very similar to our approach.
Okay, so you set out to recreate something like ZNB wave and also something like PCA, maybe take the best of the two worlds. And how did you manage to do that exactly?
Well, we basically just focused on the fact that PCA is really assuming implicitly a Gaussian or normal distribution for the errors. And we really were convinced based on the negative control data that something like either a Poisson or a negative binomial, like a discrete distribution, would be more appropriate for an error model. So we just simply said, well, what is the optimization problem that regular PCA solves? And can we just swap out the Gaussian likelihood with the Poisson likelihood? And it's really pretty straightforward if you have familiarity with generalized linear models in a regression context, because linear regression is a generalized linear model where you assume a normal distribution for the errors. And you can have a Poisson GLM where you have a Poisson likelihood. You can have a negative binomial GLM. So we just basically took that exact approach and transferred it from the regression context to the unsupervised dimension reduction context, and spent a lot of time trying to figure out how to optimize that objective function. I do not claim that we have found the perfect, best algorithm for optimization, but we found one that seemed to work well enough on our assessment data sets and on our simulations and things like that. So, we call it GLM PCA, generalized linear model PCA or generalized PCA. And after we implemented it, I was informed by, I think it was actually Dr. Engelhardt, Barbara Engelhardt, who is my current postdoc advisor. I think she was the one that told me, oh, by the way, this is really similar to something that's been done years ago in the machine learning computer science literature. So I went back and cited, I believe Collins was the author. I don't remember the year, but he called it exponential family PCA. We're not exactly copying what he did because he only used a natural link function, so he was not able to do negative binomial likelihoods, which don't usually use the natural link. And also his method did not allow adjustment for covariates or offsets or that sort of thing. So we brought some of those ideas over from ZINB Wave. And, we're really not trying to say that we were the first person to invent generalized PCA, but more our paper is really more trying to advocate for people to just change the mentality from how can we convert our data into something that looks Gaussian, and instead focus on let's actually model what's going on with the errors and the biases rather than trying to normalize them away. I think that's really the main message that we want to put out there, rather than claiming that we were the first ones to come up with some amazing dimension reduction. But we're happy for folks to use our method if they like it and give us feedback. It's available as a R package on CRAN and also a Python package on PyPy. And we're currently working on a bioconductor package that will allow it to interface with single-cell experiment objects and things like that.
Perfect. And if people want to know a bit more about what's going under the hood there, but maybe are not familiar with the GLMs, generalized linear models, can you give a brief primer on what that means?
Sure. Well, first of all, I would really like to recommend a textbook that I've found helpful for learning about GLMs, which was Alan Agresti. It's called Foundations of Linear and Generalized Linear Models. Agresti's known as a very good teacher. I believe he's retired now, but this is just a great textbook. You do need to have some familiarity with probably calculus and linear algebra to grasp most of it. It's a good mix, I would say, of theory and application. But my description of GLMs would be they sort of have three primary components. One is just the likelihood, which is it really is essentially the loss function that you're going to use for your optimization. So how do you quantify the difference between the data and what your predictions of the data are? Do you just take the mean squared error, or do you use some other kind of possibly asymmetric divergence metric to quantify this discrepancy? And this notion of a loss function is really more popular in the computer science literature, the machine learning literature, but it's actually an exact one-to-one analogy with the statistical concept of a likelihood and maximizing the likelihood. So, for example, I've already mentioned that the Gaussian or normal distribution likelihood, maximizing that likelihood and finding the maximum likelihood estimator, which is a statistical estimator for the underlying parameters of the distribution, is exactly equivalent to minimizing the mean squared error between your predicted mean of your data and the data itself. And in machine learning, there's also a notion of Kullback-Leibler. I'm not sure if I'm pronouncing that correctly.
Yeah, or Leibler.
Kullback-Leibler divergence, which is a popular metric, or divergence. It's not actually a metric in mathematical sense, but it's sort of a divergence, which is like a distance metric that doesn't have to be symmetric. But that is popular in, for example, non-negative matrix factorization. And it's exactly equivalent to maximizing the likelihood under a Poisson error model. So anytime you hear somebody talk about a loss function, cross entropy loss in neural networks is just a Bernoulli-Logit likelihood. So I find it helpful to think in terms of probability distributions and hierarchical models, but that may just be from my statistics training. But if you prefer to think of loss functions, that's certainly another conceptual framework. But the bottom line is, whatever you want to call it, the resulting estimator or the resulting parameter value that you saw from your optimization should be the same. Of course, these problems are often non-convex, and you may not get the actual exact same solution, but the problem trying to be solved is the same problem.
Right. So I think at different times in your method development, you want to take different perspectives on this, right? So once you have the likelihood, then you go into this optimization perspective where you just treat it as a loss function. You find the set of parameters that maximize the likelihood. But when you are designing your model, you probably take the statistical perspective, right? Because if you just think about this as a loss function, well, then how do I choose my loss function? I would choose something convenient like a minimal sum of squared errors. And if you take the statistical perspective, then you sort of think about what process might have generated the data and therefore what statistical properties the data might have.
Absolutely, yeah. So that's really where I think, and this is something I want to give credit to my advisor, my PhD advisor, Rafa Irizarry. He really encouraged me to try to get into the data and make histograms, and pretty much never went to see him in his office without making a lot of graphs. And a lot of statisticians and even machine learning people, unfortunately, there's sort of an incentive to try to come up with the most complex model and try to impress people with a lot of fancy math stuff, which is important. Theory is definitely important. It's the ground that we stand on. But when you're dealing with you have a lot of data, a lot of public data like this genomics. We always, can go to Geo and download all kinds of stuff. Why not look at it, and why not make some histograms and try to use exploratory data analysis to inform our choice of loss function or our choice of likelihood. And I do find it easier to look at a histogram and say, oh, this looks like a Poisson distribution. Okay, so I should probably use a Poisson likelihood, and that corresponds to, this mathematical formula, and now I have my loss function and I know what I want to optimize. So that was sort of the approach that we took, rather than starting from a highly theoretical perspective and coming up with the most complicated model we could and proving all kinds of theorems. We try to be, I believe the term is problem forward. I'm probably saying it wrong. It's not problem forward, solution backward, but the point is that you start with a problem, you start with some data and explore it and figure out what's going on with an open mind, and then bring some statistical skills or some machine learning skills to bear on the problem, rather than trying to create a new method and then go looking for a problem to solve with the method, which may not even exist.
Okay. So we sort of addressed the generalized part of generalized linear models, and that's the part where you're free or almost free to choose the likelihood or the distribution for your data. And I think you chose the negative binomial distribution for your UMI count data. But let's transition to the linear part. So why is there a linear part there?
Sure. Well, I've only mentioned one of the three components of a GLM. So the first component is the likelihood. The second component I'll just quickly mention and gloss over is the link function, which connects the mean to the last part, which is the linear predictor. So the linear predictor in a regression model is you have a bunch of covariates and you have your regression coefficients, and that's the covariates are usually like some big matrix X, and the coefficients are a vector beta, and X beta is a linear predictor. And typically that linear predictor is going to be real valued because your covariates could be any kind of variable. So, and the coefficients can be any real number. So that linear predictor is could be positive, negative. It's not discrete. It's just continuous. But then if let's say you're using a Poisson or negative binomial likelihood, what you want to do is have this linear predictor be the thing that determines the mean of that likelihood. So the problem, you can't directly equate the two because X beta, it could take on any number, but the mean of a negative binomial and the mean of a Poisson has to be greater than 0. So you need to have a function, a deterministic function that maps from the reals to the whatever the space that the mean can take on. And for Poisson and negative binomial, it's convenient to use just the exponential function. In fact, that is what's called the natural link for the Poisson. Using the natural link basically just simplifies a lot of the computations. And sometimes it makes the interpretation easier, but it's not strictly necessary. Negative binomial, the log is not, or the exponential is not the natural link function, but we use it anyway because it's more, makes things a little more interpretable, and actually it solves a problem of the dispersion parameter being intertwined with the link function, which I don't want to go into. But it turns out if you use a Gaussian likelihood, then the link function ends up just being the identity function, because the mean of a Gaussian can be any real number, and so you don't really need a link function for that. But for any other likelihood, gamma distribution or Bernoulli distribution, you need a function that will sort of squash the real valued linear predictor and map it to the mean. So I do want to mention one other thing about GLM PCA. So I met someone prior to developing this method at a conference. His name is Justin Silverman, and he is a very talented researcher in the microbiome field. I believe he's starting as an assistant professor in Penn State. But I had a great conversation with him that really inspired me to think about the idea of a multinomial model, which we haven't really gone into in much detail, but I think due to time, I'll probably just say we do discuss the properties of multinomial models in detail in our paper, but it's not like we just went straight to the Poisson and straight to the negative binomial. We really wanted to we really were convinced that the true distribution of these UMI counts is actually multinomial. Even though it looks like a Poisson, the data really only contain relative information rather than absolute. So if you have one cell where the total count is 100 and another cell where the total count is 500, you can't really directly compare the counts of a particular gene across those cells. So that's really the reason why people are doing normalization. They're converting from absolute units to relative units. And so we wanted to include that notion of modeling the relative data rather than absolute counts in our model, which we basically claim is GLM PCA. We claim is an approximation to a multinomial model. It's not a true multinomial model. And I would list latent Dirichlet allocation as an example of a true multinomial model. But we just found that the discrete approximation to it that was a lot more computationally tractable, we felt was fairly reasonable for single-cell RNA-seq data. It may not be reasonable for microbial data, which we have not investigated. That's a fascinating topic, I think, that others have looked into. I believe Riso and Levi Waldron had a paper where they compared some of the techniques from single-cell and metagenomics to see whether tools from one field could be transferred back and forth to another. So that's something that I would encourage folks to study. And I think Tom Quinn was on your program and talked about compositional data. So that's sort of the multinomial is a discrete compositional likelihood if you really care about that relative data versus absolute data.
Right. So you could say somewhat correctly that in the same sense that a Gaussian distribution or normal distribution is an approximation to, say, Poisson distribution, in the same way you can approximate the multinomial distribution with a set of binomial distributions. This is like a tower of approximations, right? You can approximate multinomial with binomial, then you approximate binomial with Poisson, then you approximate Poisson with normal.
Exactly. That is exactly what people are doing when they apply normalization. And normalization is actually maximum likelihood estimation of multinomial parameters. The parameters are the proportions of each gene within the overall counts and just the total count. So in terms of math, the approximation of a multinomial, which is a joint distribution over all the genes, which is the reason it's so computationally intractable, by independent Poisson distributions, which is just modeling the error for each gene separately and ignoring the big correlation between all of them that they have to sum up to a constant. That's a reasonable approximation if the total count is very large and if no one gene totally dominates over all the others. So in my experience, you would pretty much never expect to see one gene with more than even 10% of the total counts. I mean, certainly having one gene that constitutes 50% of the total counts in a cell seems pretty far-fetched. That may not be a far-fetched assumption for microbial data. I could imagine that some of those data, you could have one microbial strain that constituted more than 50% of the sample. And once you get to that level, the intrinsic, it's an anti-correlation actually between all of the other strains and that strain starts to really matter and starts to distort the results if you ignore it. But I think with the single cell data, and probably Silverman might, I think he would disagree with me on this, but I'm just going to say what I think. I think it's okay to assume the Poisson or the negative binomial approximation. I don't think it's okay to assume a normal approximation to the Poisson because there's too many zeros and ones and the data are too discrete. I think that approximation only works if not just the total count is large, but all of the counts are large. So with bulk RNA-seq, it might be okay, but with single cell, that's where I draw the line.
Okay, and in the remaining minutes, I'm really interested in this secret sauce behind GLM PCA, which is the combination of the two, right? Because GLMs have existed for a long time, and you named a relatively recent textbook. There are certainly like older textbooks on GLMs, on generalized linear models, and PCA has existed for a long time. But you sort of combined the two, and you moved GLMs from a regression problem to this dimensionality reduction problem, or in machine learning they call this unsupervised learning problem. So what's the secret sauce there?
Well, you have your loss function, right? So that's a function of the data and the mean, the predicted mean, and then you can write out that function. Then you just say the mean is defined as the link function applied to the linear predictor. And the linear predictor is just these two low-rank matrices, just like any other linear dimension reduction. I call them U and V. So if you have a high-dimensional data, you could have one matrix that has the big dimension being the number of features, and then a small dimension being the number of latent factors, and then a separate matrix, which is number of latent factors by number of observations, and then sort of the outer product of those two low-rank matrices gives you a matrix that's the same size as your data, which is the linear predictor that then is a function of that then is passed through the link function to give the predicted mean. So it's just like any other, people doing neural networks or whatever. You have this notion of layers, and essentially this could be thought of as a one-layer neural network where you don't have any activation ReLU functions or whatever. You just have a linear predictor, and then that thing passes through a single link function that goes into the loss. And it certainly would be possible to solve this using a PyTorch or any number of automatic differentiation libraries, but we just went ahead and solved it by hand and implemented it in plain R and plain Python. So I'm sure if other folks out there are interested in optimization, they'd be welcome to take the idea of GLM PCA and try their own implementation. And I would not be surprised if they could come up with a better implementation than what we did, because we're more statisticians than engineers. And we actually do hope to improve it ourselves. If nobody else beats us to it, we're hoping to make it work, with many batches of data, so it doesn't have to load the whole data set into memory and things like that, which would facilitate working with larger data sets.
So I think the connection to GLMs is that in a GLM, inside this link function, you have a linear combination of your parameters and your predictors, right? Your covariates. And what you did in order to make it unsupervised is to make both of them your parameters, right? So you don't have any covariates, but you say, we have parameters, we have covariates that are also parameters that we also hope to infer via this optimization.
Yes, that's exactly right. So in a regression context, we have the matrix X of our covariates and another either a vector or a matrix if it's multivariate of unknown parameters. But the covariate values, so those are fixed. But in a dimension reduction context, we actually don't know the value of X either. So you sort of have two different types of parameters that are multiplied together to give the result. And that's what makes it non-convex. That's what makes it so hard to solve. If we did know the latent factors, it would be easy to solve for the loadings. And if we know the loadings, it would be easier to solve for the latent factors. But unfortunately, we don't know either. And that's why we have to solve this really difficult non-convex optimization problem.
And so another popular dimensionality reduction tool for single cell data, which we also discussed on this podcast, is SCVI. So do you have any thoughts about how your method compares to SCVI and when one might choose GLM PCA versus SCVI or vice versa?
I love SCVI. I think it's a great idea. I think they've done a great job. Lopez and et al. I know Adam Gayoso also worked a lot on it. I met him recently. They've done a great job, and I would recommend that method. I think the reason why the distinction between our method and ZINB wave and SCVI is a nonlinear dimension reduction. It's based on a variational autoencoder, and so you get nonlinear factors. There's no orthonormal rotation. There's no necessary ordering of the factors and decreasing magnitude of influence. You can certainly encode more information with fewer dimensions with a nonlinear manifold compared to a linear one. So I think it's really dependent on what your goal. If you're open-minded about a new type of dimension reduction, which I think would definitely be more principled than something like TSNE or possibly UMAP. I think SCVI could be a great choice. However, if you are working with collaborators who really want to be able to interpret things in a manner very similar to PCA, if they're accustomed to looking at PCA plots, they want to know about things like variance explained and that sort of thing, I think our method could be more appropriate in that context. Also, I would just mention that Svensson has sort of come up with a way of hybridizing the SCVI method with a linear dimension reduction. So he had a small preprint called linearly decoded autoencoder or something like that, and basically replaced the nonlinear decoder part of SCVI with a linear decoder, which is interpretable in a way very similar to ZINB wave or GLM PCA. So there's just a lot of great methods out there, and I think it's good that folks can choose whichever one is most suitable for their team and their collaborators. And ultimately, we're all trying to make it easier to analyze these data and come up with ways of eventually curing diseases. So that's one thing that I'm very grateful to have the opportunity to be in this field.
Cool. Well, it was a very interesting conversation, and thanks for being on the podcast.
Thank you, Roman, and I look forward to hearing more episodes from your future guests.